Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing IIT JAM question paper for the subject Biotechnology conducted in the year 2020. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of IIT JAM Biotechnology being discussed, you can go to my channel Omni Bio Entrance. In that, see for the playlist. In the playlist you will find IIT JAM. In that you can find previous year question papers of IIT JAM being explained with the answers. Then coming to multiple select questions, MSQ. Which of the following is or are auxins? 1. Naphthalene acetic acid, NAA, is an auxin. Indol 3-butyric acid, again an auxin, IBA. 2,4-dichlorophenoxyacetic acid, C, again an auxin, 2,4-D. Indol 3-acetic acid, IAA, again an auxin, so D. So A, B, C, D are all auxins. Now, which of the following is true about photosynthesis? In C3 plants, the first organic product of carbon fixation is 3-phosphoglycerate. Yes. In C4 plants, the first organic product of carbon fixation is oxaloacetate. Yes. Crassulacean acid metabolism occurs in succulent plants living in arid conditions. That is also correct. Oxygen is generated from carbon dioxide. No, oxygen during photosynthesis, it is generated from the photolysis of water, not from carbon dioxide. So D is not correct. So the statements A, B, C are correct. Then next one. Which of the following is or are involved in activation of cytotoxic T cells? So MHC1 molecules, yes, they are involved because MHC molecule display the peptide fragments from infected cells or tumor cells, so that the T cell receptor, that is TCR, it can recognize this peptide which is being presented on the MHC1 complex, and it will lead to the activation of cytotoxic T cells. So C is also correct, T cell receptor. Whereas FCR, it is involved in antibody dependent cellular cytotoxicity, not in cytotoxic T cell activation. Then CTLA4, that is cytotoxic T lymphocyte antigen 4. That one, it inhibits T cell activation. So B and D are not correct. Then next one. DNA and RNA are acidic in nature due to the presence of phosphate groups. C is the answer. Phosphate groups are responsible for the acidic nature of DNA and RNA. Because of this phosphate group only, DNA and RNA are having negative charge also. Then next one. Protozoa or unicellular? Yes. Multicellular? No. Eukaryotic? Yes, they are unicellular eukaryotes. Protozoa. So answer is A and C. Then which of the following is or are correct? Both glucose and fructose have the same molecular formula. Yes. The positions of the oxygen and carbon differ in the structure of glucose and fructose. Yes. B is also correct. Both glucose and fructose have same physical properties. That is not correct. Physical property means the glucose is white, fructose is yellow, glucose is dextrorotatory, fructose is levorotatory. Then glucose is less soluble in water than fructose, and glucose is having higher viscosity than fructose. So all these are the various physical properties. So they are not the same in both glucose and fructose. So C is not correct. D. Both glucose and fructose are monosaccharides. That is also correct. So A, B, D is the answer. Which of the following gases function as signaling molecules in the human nervous system? So nitric oxide, yes. Carbon monoxide, yes. Argon and helium, they are inert gases. So they are not involved. Then numerical answer type questions. First one. The generation time of E. coli is 20 minutes. Okay. If there are 10 raised to 6 E. coli present in an exponentially growing synchronous culture, then the average time required to obtain a final population of 4 into 10 raised to 6 E. coli is. So they have given initial population of E. coli, that is 10 raised to 6 or 1 into 10 raised to 6, and final population as NT, that is 4 into 10 raised to 6. So number of generations is given by the formula N is equal to log NT minus log N0 by 0.3. Substitute and you will get number of generation as 2. Now each generation is of 20 minutes. They have given generation time is 20 minutes. So for 2 generation it will be 40 minutes. Okay, number of generation is 2. So for 2 generation it will be 40 minutes. Then next one. The volume required to prepare 350 ml of 1x buffer solution from a 50 times concentrated buffer stock solution is. For such kind of questions, you have to use the formula M1B1 equal M2V2. M1B1 is what you have as stock, your stock solution, and M2V2 is how much you require to prepare from that stock. So stock here is 50 times concentrated. So 50 into X. You don't know the volume. How much volume you have to take from this 50X concentrated buffer stock solution to get 1X buffer, that is 1 molar buffer of 350 ml. So 350 divided by 50 will give you 7 ml. So the answer here is 7. Then next one, a compound microscope has its objective with linear magnification of 10. In order to achieve final magnification of 100, the angular magnification of the eyepiece should be. So magnification for compound microscope is given by the formula final magnification is equal to magnification of the objective into magnification of the eyepiece. Here you don't know the magnification of eyepiece, so you give it as X. The other two you know. Objective magnification is 10 and final magnification is 100. So X is equal to 100 by 10, that is 10. Then next one. The decimal reduction time of a bacterial culture is 1 minute. If a suspension of the bacterial culture contains an initial population of 10 raised to 6 cells, then the time in minutes required to reduce the number of bacteria to 10 by heat treatment is. So you have the formula T is equal to D into log N0 minus log NT, where N0 is the initial population and NT is the final population. D is that DRT or D value, decimal reduction time. Okay. So you have to calculate the time required to reduce the number of bacteria to 10 by heat treatment. So substitute the values. Log 10 raised to 6 minus log 10 is equal to 6 minus 1, that is 5 minutes. Then next one. The median Y in the following data is. So arrange the data in ascending order. Even number of values. So how many are there? 1, 2, 3, 4, 5, 6. That is even number of values or even number in the data. So even number of values. So median is average of 2 middle values. So middle values here are 14 and 16. So average of that will give you the median as 15. Answer is 15. Then next one. A variable number of tandem repeat locus has 15 different alleles. The number of genotypes possible in a population for this VNTR is. So the possible number of genotype is given by the formula n into n plus 1 by 2, where n is equal to 15. Substitute the values and you will get it as 120. So that's it. Thank you for watching my channel. Please do like, share and subscribe if you are new to my channel. And if you want still previous year question papers of IIT JAM been answered, you can go to my channel Omni Bio Entrance. In the playlist, select for IIT JAM and you will find them there. Thank you.